Hi, everyone. Hello, welcome to this week's Single-cell and Exploration Club. My name is Julie. I'm a scientist here at BioTuring, and for this week's club, I'm going to be building on a story that our other scientist, Hazel, was using in a case study in the previous weeks. So in that week, she was examining some spatial data, and from the findings from that, I wanted to maybe bring over those findings and those same questions to see if we can see the same pattern in single cell data as well. Sorry. Okay, and so before we dive into the analysis, I want to introduce two of the features that I'm going to be demoing in today's club. The feature here, this first feature here is the cancer cell prediction tool on BBrowserX Pro. So for those of you who are not familiar, this tool is pretty useful for those who are studying tumors and for cancer biologists. So as we know, tumor tissue is composed of a lot of different cell types, some that are malignant and then some that are non-malignant. And so it's really important to be able to identify the cells that are actually cancerous, right? Actually proliferating. And so in this cancer cell prediction approach, the way this tool works is that it will predict which cells are likely to be cancerous based off of their predicted CNV score. Now, CNV stands for copy number variation, and this is when we see patterns of genes or genes that are proximal to each other, upregulated in some pattern or downregulated in some pattern. And so we can predict or infer what the CNV score is based off of a cell's gene expression profile. We use the cancer cell prediction tool on a single cell data set. And now this single cell data set is actually Visium HD. For those of you who are familiar with spatial data, Visium HD is basically you're looking at the spatial data in individual spots. And so when we took those spots, we were able to map it into a single cell format and then use the cancer cell prediction tool to predict which cells are likely to be cancerous. And then we map it back to the Visium HD spatial data here. You can see how on the periphery of the tissue here are the cells that are likely to be cancerous. So these blue areas here, the dark blue areas, are the ones that were predicted to be cancerous by the tool. And so one way that we were able to benchmark this or verify these results is within the same data set, an expert histopathologist was also able to go and look at the H&E image and actually look at the morphology of the cells and identify that, okay, the cells on the periphery of the tissue are the ones that are actually cancerous epithelial cells. And you can see that denoted by the yellow cells here. So now when we compare these two images of the tissue side by side, you can see that the predicted cancer cells match quite well with what the histopathologists have annotated as cancer cells. Okay, and so that's one way in which our team was able to approach benchmarking this cancer cell prediction tool here. And I'll show you in a little bit how you can actually run the tool on BBrowserX Pro. Next here, the second feature that I will be demoing is called pseudotime or trajectory analysis. This is particularly useful if you want to track gene expression changes across either different states of a cell or across different stages of diseases. In my case, I kind of wanted to see if tumor cells were becoming more invasive or more malignant, I guess, was my kind of biological question for the data set that I was investigating. And so I will also show you guys how to use that feature in a little bit as well. For bioinformaticians and computational biologists, we use this particular package, Monocle 3, to run the pseudotime analysis. Okay, and now jumping into what my findings are going to build upon. So here previously on episode 52, Hazel presented this spatial tissue here, which comes from lung adenocarcinoma. And you can see here how the authors have been able to segment this tissue into two major regions. The region on the left, which denotes well-differentiated tumor cells, and the region on the right, which denotes the more malignant or mucinous regions. And you can kind of see already by the morphology of the cell shape here that these cells are quite different from the cells over on the left here. Within the SCSE club, she also had specific gene markers that denoted for the different regions. So for example, you can see how the expression of this particular transcription factor, NKX2-1, is more present on the left side of the tissue, but not really on the right side of the tissue. And so this transcription factor is one for alveolar epithelial lineages. On the other hand, the right side of the tissue is denoted by the expression of HNF4 alpha, which is a different transcription factor, but this is for gastrointestinal epithelium. So remember that this tissue actually comes from lung, and so when we see transcription factors for other types of tissue, this leads us to believe that there is some sort of metastatic event happening, or that these cells on the right-hand side of the tissue are more invasive and malignant. And so the hypothesis here is that these tumor cells from the left-hand side may actually be transdifferentiating into the right-hand side region. And that's kind of the basis for my hypothesis as well, for a different single cell data set that I wanted to investigate. Okay, so introducing the data set here that I'll be using in the demo. You can find this on Talk to Data, but in this data set, again, the authors are investigating lung adenocarcinoma. And a little bit about their experimental design here. They were able to take samples from 5 different tissue types. So here they're taking lung sample from the adjacent normal tissue. And then they're also taking samples from the primary tumor as well. And then also samples from 3 different sites of metastases. And so overall, they were able to collect about 40,000 cells within the entire data set. However, for the purposes of this demo, I'll only be focusing on samples that come from the primary tumor. Some of the kind of exploratory investigation you can do with this data set is you can look at how the different cell types are distributed across the different tissue sites here. So we're comparing the primary tumor against the metastatic sites and then against the adjacent normal tissue. And maybe you can find something interesting here from this visualization. This is generated from BBrowserX Pro using the cell proportion tool. Some patterns that I see right away that is pretty typical of cancer's tissue is that there is a depletion of natural killer cells, especially in the primary tumor and the metastatic site. And you can see that here through the green population of cells. Okay, but I think more importantly, we want to focus on the epithelial cells themselves because in lung adenocarcinoma, epithelial cells are the ones that actually undergo the uncontrolled proliferation. And during a state of metastasis, they can adopt more stem-like characteristics. And this is known as a process known as epithelial mesenchymal transition, or EMT. So this is where epithelial cells will kind of transition into a more mesenchymal stem cell state and adopt those type of stem cell characteristics. All right, so let's go over what I did here first. So in this first analysis, I just wanted to see if I can identify those same spatial tumor regions in my single cell data set. So again, I'm looking to see if I can identify the tumor region and the malignant mucinous region here. Within the entire data set, I was able to recognize some major cell types, such as the epithelial cell types up here, the macrophages down here, and the T cells down here as well. And if I got a closer look at the epithelial cell population, and I used the specific transcription factor marker genes to mark for those different regions, I was able to find a pretty clear divide between cells that only express NKX2-1 and cells that only express HNF4 alpha. HNF4A. So here I was able to clearly define those two separate tumor regions and the malignant mucinous region here. Okay, and so next I wanted to characterize just the tumor region even further, because of course we can just use these single genes as a marker for, okay, this is the tumor region or this is the malignant mucinous region. But in order to get a more clear picture of what those regions look like, this is where I actually use the cancer cell prediction tool on our platform. So here on this image, what you're looking at is the distribution of the CNV scores that were predicted. And you can see that the scores that were highest are within the epithelial cell population here. So again, the cells that are in dark blue here have the highest CNV scores, and these are found mainly in the epithelial cell population. Going even further, I wanted to kind of verify these results. So here I built a violin plot where I'm looking at the CNV scores across the tumor region here and across the malignant mucinous region. And you can see here how in the tumor region, the CNV score is much higher. And this is what we expect because tumor cells are going to be the cells that have the highest CNV score compared to all of the other cell types. Right. And so this is one of the ways in which I approached, verifying these infer CNV results. Okay, even furthermore, I wanted to see what other marker genes are kind of present within this tumor region here. So I was able to run a marker gene analysis, and then some of the markers that popped up include all of these other markers, which I've listed here. Okay, so all in all, this cancer cell prediction tool was able to infer that the NKX2-1 tumor region has a higher predicted CNV score than the malignant mucinous region, right? And this is what we kind of expect. Okay, and so before we get into this next slide here, let me demonstrate how to run the cancer cell prediction tool on BBrowserX Pro. So you can find the tool located in the toolbar on the left-hand side. You open this up and you click New Analysis. The first thing you have to do here is specify the cancer type. This is important to specify because cancer types are very heterogeneous. And so why we want to specify this here is actually because if we were to use the automated reference type to help us predict what the reference would be, then that actually depends on what cancer type that is. So for example, in carcinoma, we know that epithelial cells are the malignant cells, right? And so when we choose the reference type, then the tool is going to try to choose a cell that is not an epithelial cell as the reference point of the analysis. So for example, I'm going to choose carcinoma because this data set comes from lung adenocarcinoma. And for the reference type, I'm going to choose the automated method. And what this will do is it'll probably look for some immune cell type signatures to be the reference point. But you can also choose the reference type manually as well. So if you were to choose manual here, then you can choose, for example, B cells as the reference point because B cells are likely not going to be the malignant type of cells in carcinoma. Okay, so I'll go back to automated. Then I can click Run Analysis here. And then I can quickly go over some of the visualizations that you can get from this cancer cell prediction tool. So the first visualization here is your UMAP. You can see here how it's divided into the cancer cells and the normal cells. Over here on the right-hand side is actually the distribution of the predicted CNV scores. And this threshold here actually determines the cutoff for what is to be considered a cancerous cells. So for example, if I were to move this threshold up a little bit more, then cells with this CNV score or higher will be classified as cancer cells. Right. And by default, we actually use a specific algorithm here called Otsu's method to determine where the default cutoff would be for what would be considered a high CNV score or a likely cancer cell. You can refresh this to reset the cutoff here, or of course, you can also move this threshold to change it manually as well. Okay, moving forward, we also have a heat map here. So this heat map will show you kind of where the CNV events are likely to be happening. So for example, let's choose an area like this, for example, in chromosome 6, you can see in the cancer cell population, there's some depletion of the gene expression within this area of chromosome 6. But for the rest of the normal cell population, most of that gene expression is actually upregulated, right? And so this is where you can see that difference between the CNV or the gene expression. Okay, and then lastly, we also have a phylogeny tree here. So this basically allows you to track how your cancer cells are evolving and in which population. So you see here how as we track down the groups of cells, so each individual node represents like a group of cells within the UMAP here. You can see within this branch here, the CNV score is the highest down here. And if you click on a node, what it'll do is it'll enlarge the node, but also enlarge the points on the little UMAP preview up in the right-hand side. Okay, and so that's how you can interpret some of these visualizations that are output in the cancer cell prediction tool here. All right, and so moving back to our results, another thing that I want to demo here is also BioVinci as well. So for those of you who are not familiar, BioVinci is our other platform for you to customize some of these figures and prepare it for scientific publications. So you can do that by exporting a lot of the charts from BBrowserX Pro. So here, if I go into visualization, basic charts, and I want to build the violin plot as you saw earlier. Click this, and then I want to see the distribution of the CNV scores. This one, I believe. Okay, we actually have an export to BioVinci button located here. And once that's ready to open, you can click open in BioVinci, and this will open a new tab. And while we wait for this to load, we received one question in the Q&A from Zania. So she asks, are these recorded and how can we access the previous episodes? So yes, all of our SCSE clubs are recorded. You can access the previous clubs on our YouTube channel. So you can look up Bioturing team, or maybe my colleague can also drop the link in the chat as well. So we always have recordings of our past episodes as well. This recording will probably be uploaded next Monday, so stay tuned for that. Thank you. All right. And so basically, yeah, let's wait for this to load a little bit more. So what this is doing is it's loaded in the BioVinci application now, as you can see. And now we've entered the editing mode. So things you can do in editing are, of course, you can change the font here, for example. Fonts here. You can also change the font type. And also it's important to change the color as well. So I want to change this to maybe an orange, and maybe this one to maybe a darker blue. Okay, I can also resize if I'd like. And then if I want to get rid of the grid lines here, I can go over to the axis lines here, and I can untick some of the grid lines or the ticks. Maybe let's show some Y ticks. And then we also have some more advanced editing as well. So for example, you can edit either the violin body, the box, or the data inside as well. For those of you who have not explored this BioVinci app recently, we also added this view data feature here. So you're able to view this data along with the cell barcodes and download this data as well. So that if you'd like, you'd have access to the gene expression for each of these individual cells. Okay, and once you're happy with the image here, you can, of course, export this. Let's save this, and then I can show you all of the different exporting features that we have. So you can export it as a PDF, SVG, PNG, or JPEG and put it into your publication if you'd like. Okay, so that summarizes some of the figure customization that you can do with BioVinci and the violin chart here. So let's move on to the next analysis. So here I have the question, okay, do we observe the transdifferentiation event in lung? In other words, can we see whether the tumor region is actually kind of transforming into the more malignant, mucinous, invasive region within the single cell data? And so to demonstrate this, I'll go back over here to our pseudotime. Can open up the pseudotime tool here. And one thing that's important to consider here is you want to select the correct UMAP that you want to use within the analysis. So in my example, I want to only look at those two regions that I'm interested in. So I'm only interested in the HNF4 alpha positive region and the NKX2-1 region. So I'll select those two. And then pick subcluster here. Then whenever you subcluster, you can also choose to run a new UMAP or TSNE as well. In my case, I had already run it previously. Here already. Okay, and so for the pseudotime, then I can just click run. And so in doing so, this will take maybe a minute or two to run. I can show you a results page here. So when you load in the pseudotime for the first time, what you're going to want to do is you choose a root node, or essentially choose the starting point for the pseudotime. So for example, what I noticed right away from the UMAP, let me enlarge these points so you guys can see a little bit better. What I noticed right away from the UMAP is that there's a cluster of cells outside here that kind of cluster away from this larger cluster here. So this leads me to believe that, okay, maybe the starting point is either up here in the small cluster or down here in the larger cluster. You can also see that for my regions that I'm defining, they're kind of dispersed throughout this cluster. So it's kind of hard to see where the starting point for the tumor region would be, which leads me to believe that perhaps the pseudotime analysis here might not be able to, I guess, tell me anything that might be biologically relevant. So let me explain that in the slides here. So here you can see how the tumor region and the malignant mucinous region are not really well defined. They're kind of dispersed within each other like this. And so even if I were to choose a root point anywhere within this trajectory analysis, I don't think the results from this type of analysis would have told me anything biologically meaningful. And that's okay. Sometimes tools are not always going to be relevant to your analysis, because the pseudotime or the trajectory analysis is very heavily dependent on what the UMAP or the TSNE will look like. So if you were to see kind of a more clear separation of these two regions, then maybe you'll be able to kind of interpret the results more meaningfully. So again, just to reiterate, it was really difficult to distinguish between these two tumor regions and the malignant mucinous region. And so performing pseudotime analysis didn't really output any meaningful results for me. Okay, and then in this last analysis here, I wanted to kind of approach that question or that biological question in a different way. So still I wanted to see, okay, do we observe any differentiation event in the lung? And so I can answer that question using cell-cell communication actually. So you can see how here in the UMAP, the two regions are kind of dispersed quite close to each other. So maybe there's still some communication event or interaction going on within these two regions. So with cell-cell communication, I was able to identify that these two regions are possibly communicating with each other through this AGRN or agrin pathway. And so you can see here, I've denoted that AGRN is implicated in lung adenocarcinoma progression. And this other paper here reports on that result and does a deeper dive into this hypothesis here. And what you're looking at here on the left-hand side is one of the visualizations that are output from the cell-cell communication tool on BBrowserX Pro, where you're looking at the expression of the AGRN ligand and the expression of the DAG1 receptor. And you can see how the expression of these two genes are quite specific to the tumor region and specific to also the malignant region. And these two regions are communicating and interacting with each other via the ligand receptor pair here. Okay, and so those are some of the interesting findings that I was able to kind of collect from this analysis and build upon from the Spatial X SCSE previous SCSE Club episode. Okay, and so that ends today's club. Thank you everyone for joining today. I left this little last note here because of course this type of case study can always be expanded upon. I think it'd really be interesting to see if you can find the same results across multiple studies, and you can do that by utilizing talk to data. So you can find more lung adenocarcinoma studies and try to, answer the same questions that I have posed in this club. So my colleague just sent out a poll as well. We'd like to gather feedback on the platform here. And so any feedback that we gather from here greatly helps us improve our platform in the future and also helps us, direct SCSE clubs in the future as well. So please, if you have time, fill out this poll and it'll be super helpful to us. Okay, I'll give it a minute or so for everyone to finish answering the poll. Otherwise, we're open to Q&A as well. All right, I think that's good for today. So thank you everyone again so much for joining today's SCSE Club. Our next SCSE Club will happen in 2 weeks, so hope to see you then. Thank you.